Welcome back. In our introduction, we had mentioned that immune cells are of two main types, lymphoid cells and myeloid cells. Today we are going to look at myeloid cells. So myeloid cells are of three categories. We have granulocytes or polymorphonuclear cells, which are three: neutrophils, eosinophils, and basophils. Then we have mononuclear cells, what we may refer to as monocytes or macrophages. Then we have dendritic cells. Some people classify dendritic cells as macrophages, but in some cases it is discussed on its own, although its functions are mainly the same as those of monocytes or macrophages. So we are going to start by looking at this first group of myeloid cells, granulocyte. I'll first look by features of granulocyte. These are general features. We have three granulocytes, but these are general features, and you can find them all in one cell, one type of cell I mean, or some cells may just exhibit a few of these properties or features. So let's look at them one by one. They are polymorphonuclear cells. Their nucleus have more than two lobes. So there are some with two lobes, there are some with three, there are some with four, and there are some with five. Initially, they were called multinucleate because the light microscope then could see the lobe nucleus as more than one nucleus. Another thing, they are short-lived. Something like B cell or T cell are memory cells which can live for years once they are activated. They are the majority of white blood cells, forming around 67% of white blood cells. They are motile. That means they can migrate from one part of the body to another, like from blood, vascular system, to tissues and vice versa. So they are also phagocytic. They are phagocytic, thus they engulf foreign bodies and destroy them. Since they take part in innate immunity, they have no memory cells and they are not specific. But in their movement, as we say they are motile, they are attracted by chemicals or what we call chemotaxis. The chemical produced attract them to area of infection. We have said there are three types: neutrophils, eosinophils, and basophils. Let's start by looking at neutrophils. We look at the properties. Neutrophils carries almost all the properties. There are some properties which are functions which are never mentioned that will be seen here. So these cells will carry out what we call inflammation. They carry out cell cytotoxicity apart from phagocytosis. So let's look at this cell we refer to as neutrophil. The structure we have described much earlier. So let's just go through these properties and see what they carry from here or leave out. So they are most numerous. In fact, 6 to 7% of white blood cells. They live for a very short time. I didn't talk about that. They are short-lived. I mentioned it, I remember. I compared with the memory cells which live for many years. So they are phagocytic, non-specific, without memory. They have granules That's why they're called granulocytes. These granules carry chemicals which kill microbes. So they are microbicidal molecules. They enter infected or injured tissue attracted by chemotactic factors. They carry out cytotoxicity but aided by antibody. This one is a process I explained earlier, but we'll still talk much about it. So just know they carry out antibody-dependent cell-mediated cytotoxicity or antibody-dependent cell cytotoxicity. Dead neutrophils form pus. When they migrate to area of infection, they fight the antigen and they also die. So the dead neutrophils form pus. In female, I cannot draw now, but maybe I can explain this in class or much later. You see that the nucleus is lobed. Like this neutrophil must have come from a blood from a male human being. If you get a neutrophil from blood of a female, then you will see some projection in any part of the lobes, straight projection with a rounded tip. It is called a drumstick chromosome because it resembles the stick we used to beat drums. So we know that nucleus contain chromosomes. So this is a projection of chromosome from the nucleus called drumstick chromosome. It only found in neutrophils isolated from female blood. Then let's turn to eosinophil. We've talked much about the structure, you can see here, but the nucleus is 2 or 3 lobed. It forms a perfect sphere compared to this one. And the granules appear much bigger. So this cell is specialized mainly in inflammation and cytotoxic function. It specializes in inflammation and cytotoxic function. So it produces chemicals from the granules and secrete those chemical enzymes or chemicals outside the body, which it uses to kill other cells, infected cells, or parasites. These cells are just single celled, but they're able to kill parasite, which is multicellular, because a good number of them come close to the parasite and secrete those chemicals and they kill the parasites. So they are not as many as neutrophils. They make 1 to 4% of total white blood cell population. The granules are having chemicals which I've said are used to kill even parasites. So their role is in parasitic defense and asthmatic allergy. That is inflammation. So let's look at basophils. Basophils also specialize in inflammation, which is called type 1 hypersensitivity. This one we will not discuss, but we'll discuss it later. We'll look at defects of immune system. So it has receptors for IgE, that is immunoglobulin class E. This IgE, together with antigen, touch on the cell and causes type 1 hypersensitivity. Details we'll see later. So the nucleus has 3 lobes as seen. It can also form S-shaped. Then we look at mast cell, which we say is a form of basophil but in tissue. The granules produce histamine and heparin, which are mediators of inflammation, type 1 inflammation or hypersensitivity. So it looks a little bit different from the basophil because the nucleus seems not to be lobed and it has granules which are much bigger with the projections which I should have earlier. Then we go to monocytes. Monocytes are not granulocytes. They are cells which are much larger than granulocytes. And very amoebic. They are very active phagocytes. You can see the pseudopodia. Very active. So they are larger than phagocytes. They have large nucleus which is not lobed but can be indented like this one here. So can be indented or can appear around depending on which cross section is taken. The other thing about these cells is that they have very few and small granules. That's why they were called a granulocyte. So under electron microscope, the cytoplasm is very clear. But under light microscope, the cytoplasm is very clear. But under electron microscope, you see organelles, which apparently is missing in the granulocytes. Most granulocytes have reduced organelles. Their parts are taken by high concentration of granule lysosomes. So they have many lysosomes. Those I'm talking about granulocyte and reduced, other organelles are reduced. But these ones have few small lysosomes, but other organelles are very prominent. So that's the different structure between monocytes or macrophage and the granulocytes. So let's go back and look at the characteristics after we have looked at the cells. So they comprise 5 to 10% of white blood cells. Short lifespan, large leukocytes, they are very large. So they are oval or kidney shaped, have single nucleus, abundant granular cytoplasm. They mainly serve as precursor for macrophage. So their function and that of macrophage is the same. So what about macrophage? Macrophage and monocytes which are in the body tissues. So they can be permanently residing in the body tissues. They are called resident macrophage. Or they can be recruited during infection, attracted by chemotactic substance called recruited or elicited macrophages. Other macrophages are here. So there are different macrophages depending on where they are found and where they work. So what about their functions? The major role of monocytes is antigen presentation. They process antigens and present it to T cells, activating them or causing them to act in a given way, and therefore destroying foreign material. They also are phagocytes. They are phagocytes. They also kill by cytotoxicity or by producing chemicals which kill microbes. So microcidal or microbicidal chemicals. They produce monokines like interleukin 1 and therefore regulate the immune system. They also contribute to inflammation and repair of body tissue. So those are the functions of uh, monocyte or dendritic cell. Their functions are the same, monocyte or macrophages. Then let's look at the last myeloid cell. This is dendritic cells. Dendritic cells, some people classify it as a form of macrophage found under the skin or the wall of the gut. So they are there, and their function is also antigen presentation. So they are specialized antigen presenters. So they process and present antigens. So like macrophage, they also produce interleukin 1, so they regulate the immune response. So they process antigens. And since they present antigen to T cells and produce interleukin 1, they control differentiation of T lymphocytes. So their main function is antigen presentation. They are called professional antigen presenter, and therefore they use that to regulate the immune system in various ways. So they are called dendritic cells because they are projections which look like dendrites of nerve cells. Thank you. That's the end. Next time we look at lymphoid cells.